All right, cool. So welcome everybody to our 10th iteration of the CMFI Mass Spec Seminar. Yeah, it's a super great pleasure to have today Efi and Axel here giving the lecture on pyOpenMS, and basically all in the realm of building like a data analysis workflow that is that fits basically like in a Python script or like in a Jupyter notebook or so on, which I think is quite useful when you're like at the more advanced stage of like metabolomics data analysis and you want to build something that you can use for like routine analysis. So particular for, I guess, high throughput applications, core facilities, or I don't know, power users with a lot of projects going on. I think this can be like super useful. And yeah, so it's great to have Efi and Axel here, just to give you like a little background. So Efi did her undergrad in chemical engineering at the National Technical University in Athens, before she then moved to Copenhagen, to the Technical University of Denmark, to do a master's in biotechnology. And then she apparently liked it in Copenhagen, so she stayed for her PhD. Then also at DTU and at the group of Tillman Weber. And yeah, Tillman has also a past here in Tübingen, so it's not surprising that Efi teamed up with people here in Tübingen, such as like Axel, who did both his undergrad and his PhD here in Tübingen in the group of Christoph Meyer, before he now also started his postdoc here in collaboration or between the labs of Christoph Meyer and Oliver Kohlbacher in the computer sciences. And for those of you who know who don't know Oliver, so his group basically like is developing OpenMS, and I guess that is the perfect background for today's lecture. And yeah, I'm very much looking forward to Efi's and Axel's presentation and hands-on tutorial on how to perform metabolomics data analysis with pyOpenMS inside of a Jupyter notebook. And yeah, without further ado, thanks again for doing this and looking forward to your lecture.
Yeah, thanks, Daniel, for the nice introduction. Yeah, hi everyone. Today we will take a look at pyOpenMS and how you can use it for your metabolomics workflows. And in this workshop, we will give you a short introduction on pyOpenMS and also on all the steps that are necessary for the pre-processing. And then we will have a hands-on session, and everybody that wants to follow can do this, open a Jupyter notebook in the browser, and yeah, go along with the interactive example. So when we talk about computational metabolomics and also proteomics, we have hundreds of experimental methods and protocols. And some of the major steps there are always the identification and quantification, as well as the statistical analysis at the end. And these methods, they are tailored to one specific experimental method. So you always have to adapt your workflows to the data you're processing. Yeah, and of course, the goal is always the extraction of maximum biological knowledge. And this is kind of the holy grail for the bioinformatics that we can see here. So the experimentalist, the biologist, he brings us a hard drive full of data, raw data. And then we have this pipeline that we don't know what's happening inside really, but we don't usually care. So we just want to push the start button and out will come ideally an Excel sheet or maybe also publication ready figures. But yeah, we know this is not the case because the mass spectrometry workflows, they are very diverse. We are dealing with, as mentioned, with proteomics and metabolomics, all kinds of backgrounds. We have different, for example, columns, LC separation methods. Then also the acquisition is quite different across all the different instruments that are out there. we might have some samples with label or without labeling. And also the experimental design that comes before measuring the data, like the sample preparation, might also play a role. And what most people want and expect is like this kind of Swiss Army knife with all the tools, so one tool to do it all. Yeah, but we think this is maybe not the right approach because it's also clumsy to use, and one single tool could maybe not be enough. Instead, maybe the solution could be a set of tools that can be combined to build highly flexible workflows. So you rather have these single tools in your garage that you want to use, and then just pick the ones that do your specific job very well and chain them together. And that leads us to OpenMS. So OpenMS is a computational library that also includes tools and workflows for the analysis of mass spectrometry data. It is completely open source and free of charge, so everybody can use it without limitations. We use standard file formats that are defined by the Proteomics Standards Initiative. And because of that, we are also vendor independent. For example, if you have thermo.raw files or pruka.d MS files, then you can easily convert them to the open format MZML with the ProteoWizard library using MSConvert. So this is a kind of overview about the OpenMS framework. And on top, we have a high level of usability and a high abstraction layer also. And down there, it is very extensible, but maybe not that easy to use. So the core foundation for OpenMS is its library, where we have algorithms for identification, quantification, visualization, and also all the data formats and data structures, like, for example, a feature map or an MS experiment are stored inside of these libraries. If you want to jump in right at the top, at the most usable section here, we also support different workflow engines, such as KNIME, Topaz, Galaxy, and Nextflow, and I will introduce them a little bit better in a minute. But there's also an option to jump in right in between. So we have these top tools that are application-specific tools that have a common interface. And this common interface just means that they share input and output file formats, and you can easily link them together. And one focus of this talk today is also the scripting capabilities of OpenMS. So we have wrapped most of the code actually for pyOpenMS in Python, and you can use that to access all the algorithms and build your own workflows. So as mentioned, the OpenMS tools are the top tools, also the OpenMS pipeline tools. And right now we have close to 200 of these. And the idea is to have one small tool that does one of the analysis steps very well. And the identical user interface is just there to ensure that we can build workflows with them and they are all compatible with each other, because they exchange the data in these PSI standards formats. And because all is in standard formats, it's also easy for us to support different workflow systems and also to work together with other tools such as Sirius or GNPS. And these are the OpenMS workflow managers I mentioned. And yeah, the goal here is just to build workflows and that are reproducible and also reusable. So in your lab, you might build one workflow that is very specific to your application, tailored towards your instruments and your experiments. And then everybody in your lab can later reuse that and open it in these workflow managers. So for example, KNIME is very good for beginners because it has a nice graphical user interface. So all the tools are these nodes here that you can put together. And it's visually very nice. It's kind of visual programming and good for beginners. But the downside might be that you cannot really do large-scale data analysis with KNIME so well. It's not that scalable. So if you have a high amount of data that you want to analyze, you're better off using something like Nextflow. And in Nextflow, you define all the steps in your workflow in one of these scripts here. And then you can take that and run it on a very powerful server or on a cluster. So if your local computer is too slow, you can do it like that. And this also makes it easy to automate stuff. And yeah, another thing is I want to mention is the Galaxy project. There you have everything in your browser, and all the OpenMS environment is already set up for you. So if you just want to take a look and play around with it, it's perfect. You can go on the Galaxy Europe server and upload your data there and then access all the algorithms. One of the downsides might be if you have slow internet connection, it's not very good, or if you have sensible data that you don't want to share on the internet or just don't want to upload. Now for scripting with OpenMS, so the core library of OpenMS is built in C++, and this has some advantages, like C++ is a compiled language, so it's very fast, and also facilitates nice memory management. The downsides might be that it's non-trivial to learn, and it has a verbose syntax. And also dependency management can be quite a challenge for the user or for the developers. You as a user, you don't care usually, but if you want to build new algorithms, it can be tough. Python, on the other hand, many experimental biologists, they know some kind of Python, or it's easy to learn at least. Maybe for some statistical analysis, or you have plotted some graphs with Python already. So the barrier of entry is generally quite low. And also the interactive scripting capabilities with, for example, Jupyter notebooks are great. Yeah, and this pyOpenMS, it basically wraps all the OpenMS algorithms for you to use. Dependencies are also easy to install via the Python package index, via pip. You can use Anaconda, or we recently added also to our tutorials website, to our read docs, always these launch binder buttons, and then you will get a Jupyter notebook in your browser with the environment set up for you. And that's what we are also going to do in this workshop. So if you want to follow along in the practical part, please visit this GitHub repository here. And what we can do then is we can launch the binder. And this should not happen. It happened now that it's not launching. I don't know, maybe I can get some feedback from you guys if it launches on your side.
No, same error here.
Okay, I guess that's because I pushed some changes recently, and it always takes a while because, yeah. I think we can then do it all locally. So
it's launching for me.
It's launching for you?
Yeah.
You want to jump in?
Oh,
no,
yeah.
I guess
so
for the people that want to follow and it's launching, you can try to launch it. And otherwise, I will just proceed in a local copy here on my side later.
Let's try later, yeah.
Yeah. All right. And while you launch it, Effie can take over and walk you through all the pre-processing steps.
Okay, so thanks a lot, Axel, for the nice introduction. And hi, everyone. My name is Effie, and I'm, as you heard already, I'm not affiliated with OpenMS at all. So how did I end up presenting today? So I have this PhD project that's called Metabolomics for Specialized Metabolites. And I aim to develop this more high throughput workflow for screening our thousands of actinomycete strains that we have in our collection in order to discover new natural products in general. So what I did so far, I selected a bunch of genomes from our strain collection. I cultivated them in a more high throughput manner. And then I ran them through our Thermo Orbitrap IDX instrument and finally ended up with 1,250 raw data. And of course, the question next was what to do with all this data, because of course, manual interpretation is impossible. So finally, I ended up building this workflow using OpenMS algorithms called Umetaflow. That's how I decided to call it. I'm not going to go through everything today. I just want to mention that I'm using a workflow manager tool exactly like Nextflow, like what Axel mentioned before, but it's called SnakeMake, and it's also very, very popular. SnakeMake has, my workflow in SnakeMake has all the OpenMS binders in a command line interface version. And then we also wrote everything in pyOpenMS language, Python language, and the pyOpenMS wrappers together with Axel using these Jupyter notebooks that are more interactive and educational. So today I will only focus on the first step that's called pre-processing. And it's the most common step used in metabolomics data processing, at least at the very beginning. So it's basically a series of algorithms that convert your raw data to a table of metabolic features. And that's what you want, like a nice clean table with features that are not noise and that are, yeah, quite interesting. So I will introduce today all the algorithms that we need that we need for pre-processing, and that you will later on use it in your workshop with Axel. So just to quickly mention the very beginning, the very initial part, the data conversion. So today you will be offered with already converted MZML files, Centroid MZML files. But in order to use OpenMS on your own with your vendor format files, you need to convert them to an open community-driven format, so an MZML, as Axel already mentioned. And you can do that through Thermo raw file parser or ProteoWizard. ProteoWizard is very popular, actually. And Thermo raw file parser, you can, of course, use it only if you use a Thermo instrument. So what do I mean when I say converting your files to your data from profile to Centroid? So if you look at the graph below, those different colors represent different metabolites. And your instrument will detect a compound mass to charge in a window of 10 or 20 parts per million approximately, depending on what instrument you're using. So it will not always measure the exact same mass to charge. It will shift a little bit. So centroiding your data allows you to reduce the information and only keep the local maximum of each range of each metabolite. So that helps a lot, making things less computationally expensive for the following algorithms. So the first step of pre-processing is feature detection, something that Thomas Puskal went really on detail in one of the Mass Spec seminars that you can take a look at. So feature detection in OpenMS requires three different algorithms. So you introduce your MZML files to mass trace detection. That's the first algorithm. And as the algorithm is named, it detects mass traces. So it gathers peaks, similars in mass to charge and moving along retention time, like shown in the figure below. And in this step, a very, very important parameter is noise threshold. So you can define a noise threshold according to your instrument and your data. In our case, we have the noise threshold at 10 to 4 because that's what our Orbitrap instrument, that's how it behaves. So anything below 10 to 4 can be considered noise. So that saves computational resources and it decreases the noise in the output. Yeah, and the extension phase of a mass trace will end when the frequency of the gathered peaks drops below a certain threshold. So then right after we have elution peak detection, and is what we call deconvolution. So each mass trace may consist of co-eluting peaks or even overlapping even eluting peaks that could be isomeric compounds with the exact same master charge, but a little bit different retention time behavior. So elution peak detection will detect local minima and maxima and separate those into two different peaks. And then finally, for feature detection, we have Feature Finder. It's an algorithm that assembles metabolite features from singleton mass traces. And what does that mean? So every monoisotopic trace is accompanied by satellite isotopic peaks, carbon-13, etc. So it makes, again, the analysis a little bit more difficult. So this algorithm fulfills one more reduction step, and it assembles compatible mass traces to metabolite features. So isotopes that belong to the same metabolite. And then finally, you have your feature files. So the next algorithm that we need to use is called map alignment. And exactly as the name says, it performs a linear retention time alignment. And that's very helpful because for any of you that has used LCMS, you know that in every single run, even or between different runs, or even in a single run, but between different injections, you have linear retention time shifts in your data. So this algorithm corrects these linear shifts by choosing one feature file as a reference file, the one with the highest number of features. Cool, and then we'll perform the retention time alignment according to that file, as shown in the figure, yeah, sorry. Cool, then we have feature linking. That's the final OpenMS algorithm that is used for pre-processing, and it clusters all the feature information from the single feature maps that we have created, the feature files, into one single consensus map. And it links features from different files together that have similar master charge and retention time. So if you look at the figure on your right, you can see two different feature maps, 1 and 2. They share one identical feature and a little bit different retention time and master charge, but quite identical. And then they also have 4 different features. So in the consensus map through feature linking, those different features will be conserved exactly as they are in the feature maps. And then the identical one will be clustered together, and the average values of retention time and master charge will be calculated. So then with a tiny Python script, we can convert this consensus map to a table of features, to a nice data frame. And if any of you use Python, I'm sure you've heard about data frames. So it's just a table, and here you have the master charge information, the retention time information, and the intensity measured in each sample per feature. And if you see any missing values or any 0 values, that means that either the instrument hasn't detected that compound, or the algorithms filtered that compound out, that feature out from the specific sample because of perhaps an intensity threshold filter. Yeah, so a few suggestions after pre-processing that I'm also using in my workflow. First of all, you can add those feature files in Sirius Adapter. It's just a wrapper of Sirius algorithm in OpenMS. And I just want to remind you that in the previous Mass Spec seminar, we had Kai Dürkop, who is from the Boker lab, that presented Sirius and CSI Finger ID. And for untargeted metabolomics, those tools are really, really helpful because they generate those formula and structural predictions. And actually, Sirius and those formula predictions, at least for my data, it's around 80% to the point. So that's pretty good. I'm also using adduct annotation. It's an algorithm called metabolite adduct decharger in pyOpenMS, and it assigns adducts and reconstructs neutral masses. So it's a further reduction step for your features. And finally, as a bonus for today, we will also look into that step in our interactive workshop. That's GNPS export. it's a few steps that we tailor together to generate all the files necessary for feature-based molecular networking, as well as ion identity molecular networking that you can forget about today. I think you're going to learn about it in the near future. But feature-based molecular networking, you have already been presented to in one of the previous Mass Spec seminars. Cool. And then we also have a tiny script for. Okay, so I'll just briefly go through that workflow actually here on the bottom right. So right before linking all those features together in the consensus map, we can use this algorithm called ID Mapper that will basically annotate all the MS2 data in your files. And that's very helpful because then you can filter out with the file filtering step all the features that have not fragmented. And that's very important for feature-based molecular networking because it only creates networks between the MS2 information. Cool. And then GNPS export will cluster all the MS2 information in a single MGF file. And that's one of the required files to submit your job in GNPS, as well as a feature quantification table in a TXT format that's also generated from that algorithm. And that's basically just a table of metabolic features in a format specifically that GNPS is expecting to receive. And then finally, the metadata table can also be generated from our just a small Python script, basically using data frames. And surprisingly, doing it manually can generate a lot of errors in my experience. So it's good if you can do it through a tiny code. Cool. And anything else, like you can build your own custom workflows. You can do anything with pyOpenMS. And you can follow this link here called pyOpenMS.read-the-docs, where we have all our documentation if you're interested. And yeah, I think with this, I can hand it over to Axel for the interactive workshop. I hope the binder worked for me. So yeah.
Yeah, thanks, Efi, for this nice introduction. And actually, I'm curious if it works now. So let's check again. And now it's launching here. But I guess because that takes a while, I will still keep to my local files here. And if you like, and it worked for you, then yeah, you can follow along or check it out later also. Just as an introduction here, or as an information about this, so we are using Python here. And if you have some basic Python knowledge, the syntax should be very convenient for you. You should know all of it. But if you don't know any Python, it's also not a problem, because you can learn about all the algorithms and all the parameters that we will set. Yeah, and maybe this is also a nice introduction to actually using Python for your research. So in this repository, we have a directory that's called example data. And in there we have two standards, two MZML files that are antibiotic standards commercially available, produced by Actinomycetes. And what we are going to do now is, from these MZML files, we will detect the features and then link them into a consensus feature map, and later also prepare the files necessary for GNPS. So what we do here in this Jupyter notebook, we have these different cells that we can run one after the other. In this first one, we import some stuff. Of course, from pyOpenMS, we import everything. OS and glob, they are for file handling. Then we have pandas, which is, Efi mentioned, is for data frames, and which are basically tables. It's a very common module, Python module. And then we also import plotly to give a nice plot of our consensus map. And we can always see if that executed with that sign here. So what we do here is we define directory path to directories for results, and also interim. And the interim directory is basically for temporary results, where we will store files that are generated during the workflow, but we are generally not interested in at the end. So at the end, we want to have our nice table of metabolic features. The next step would be the feature detection step, and for that we generate a Python list with the directory path for all our MZML files in this example data directory. So with this wildcard, we can get all the files that end with MZML. And what we then need to do is follow the 3 steps for feature detection, which is mass trace detection, illusion peak detection, and feature detection. And we have to do that for each MZML file. So we loop over them. Here we initiate an MS experiment and call it exp. And this class is a storage container from OpenMS, and it contains all the peak and spectra information that is inside of an MZML file. And we have also file handling classes, in this case the MZML file, that is used to load and also store data. So this load function, it just takes the file name, the path to the file, and an empty MS experiment, and it will load all the data that's in this file into this object. For mass trace detection, we initialize an empty Python list, and this list will be filled with mass traces that are detected by the algorithm. And the algorithm is mass trace detection in this case. So here comes the parameter settings, and this is done the same for all pyOpenMS algorithms that are out there. So we can get the default parameters of this algorithm, and then we can set values. And these values, they are more or less like key value pairs in a dictionary. So in this case, we set the mass arrow ppm to 10 because we have a high resolution OB-trap instrument. And we set the noise threshold intensity to 10 to the power of 4. And these 2 settings, they are very data dependent. So you should definitely check out what works for your data and set these accordingly. Then we have this parameters object with your custom values. And then we can introduce it back to the MTD algorithm by the set parameters function. And then it has the new parameters that you just set. And many OpenMS or pyOpenMS algorithms, they have this run function. In this case also. So this for mass trace detection, it expects an MS experiment, an empty list of mass traces that will be filled. And this is a 0. And if you don't have any idea what one of the parameters does, you can always check out the OpenMS website. And you can go to this GitHub repository. And then there's a button that says go to file. And when you do that, you can type mass trace detection, and then you will find these PXD files. And these PXD files are more or less the files where the Python code is defined. So you can check that file out. It's usually a very small one, not too complicated. Here for our mass trace detection class. And we have our run function here. So we can see that the first one is the MS experiment, like expected. The second one is a vector or a list of mass traces. And the third one would be max traces. And this is defines the number of mass traces that are added to this mass trace list. So if you set it to 100, the maximum number will be 100. If you set it to 0, it does not limit the number of mass traces, so it will put just everything that's in there. Okay, so the second algorithm for feature detection would be the illusion peak detection, and this works kind of the same. We initialize an empty list that will later contain all the deconvoluted mass traces, and we just set one parameter here, which is the width filtering, and we set it to fixed. And this means that we will filter out all the mass traces that are below a certain full width at half maximum of 1 second, and that are larger than 60 seconds. And the illusion peak detection algorithm has a detect peaks function that takes the mass traces and also the list for the deconvoluted mass traces. And the last step would be the actual feature detection step. And for this, we introduce an empty feature map. And the feature map is like the MS experiment, an OpenMS data container for features. It contains features of one specific sample. And we also need to define an empty list of chromatograms, because you can also export the chromatograms of the features in a list, even if we don't take a look at them now. So the algorithms we are using is the feature finding Metabo. And here we only set one custom parameter, which would be remove single mass traces to true. And this removes traces that don't have an isotopic pattern. So for metabolites, usually you would expect them to have 2 or 3 mass traces, depending on their isotopic patterns. And if one feature only has 1 mass trace, it's not that likely that it's a real metabolite, so we will filter it out. Then the run function of the feature finding Metabo takes the deconvoluted mass traces and the feature map. And what it does, it more or less, it fills the feature map with features that are detected. And what we also can do here is we can set unique IDs to our features. This is more or less giving them a unique name, and important also for some algorithms that will follow. So what we can do also is we can set some custom parameters like the primary MS run path as a meta value. And in this case, we will just store the name or the path to the original MZML file in the feature map. And this is to store files. And we have the file handling class for feature XML files. We just passed the file name and then the object we want to save, in this case, our feature map. And here we can take a look at them. So the feature map would be something like that. So this is an XML format. And we have here on top some user parameters, some meta values like the MZML file path, the feature count. This has 99 features. And then we also have these blocks here, which represent features. And they represent the RT, the MC, and also the overall intensity, quality value, and a charge. And also you can add meta values for your features like these. Let's actually run this cell. And it will always produce some output here. So then we can also load our feature files again. In this case, it would not be necessary. We could also pass a list of our feature maps, but just to demonstrate how you will handle files and loading and storing files, I've done it like this here. So we will load all our feature files and into feature maps and append them to a list of feature maps. For map alignment, we, as Efty has explained nicely, we need a reference map, a reference index. And this is the feature map that has the highest number of features. So what we do is we just sort our feature maps and by size, and size is equivalent to the number of features. Then we get the last one, which is the one with the highest number of features, and get the index of that one. Our aligner is here, the map alignment algorithm post-clustering. And we introduce that. We also set some custom parameters and we set a reference. And the reference map is just in our feature maps list with this index. And what we then want to do is we want to transform all the other features in the other maps. So we loop over all the feature maps that are not the reference maps. And what we have here is a transformation description. This is important because this saves the transformed data points. And you can also save that to a file and to use later to, for example, transform your MZML files themselves based on the feature map alignment. And then you can run the align function here. This does not change anything yet. The actual change is happening here with the map alignment transform retention times on this feature map, and this changes the actual retention times of the features. And then we can save them again and save them with a prefix of aligned. So they are here in this list. Feature maps not defined. I did not run the cells above. So let's do that. And then it's done. So the next step would be, as Effie mentioned, for GNPS, it's very important that we have the MS2 data that is associated with a peak or with a feature annotated as somehow annotated. And we can do that with the ID mapper class. And therefore, we need our original MZML files and we need our feature maps. So what we do here is we loop over our feature maps and our MS experiments, our MZML files, and load them into an MS experiment. And then we compare them to the feature map file name. So we always want to ID map only when the file name is the same. So if the feature map really belongs to that ML file, and then we can use the mapper and its annotate function and pass in the feature map and our experiment, and then it will annotate it. You might wonder why we have these peptide IDs list here. This is just historically in OpenMS, we can annotate MS2 data as peptide IDs very easily. And because of that, we do it like that. But it's just called peptide IDs, but we will, even if they are not actually peptide IDs. Yeah, and then we can save this feature XML file with an ID mapper prefix. And if we take a look at that, we can see that some of the features have peptide identification now annotated. And this is the MS2 information. Yeah, then we load our ID mapped feature files again. Let me run that. And load them. And one of the final steps in pre-processing is always the feature grouping. So we want to have this feature matrix table with consensus features. And the algorithm would be the feature grouping algorithm KD. And here's a new OpenMS data format. It's a consensus map. This is like a feature map, but it contains the information on features from different samples. Then we also have these file descriptions here. So in each consensus map, we need information on the original feature maps. So all the meta values, what is the name of the MZML file, and so on. So the file name. And we will get all of this from the column header of the consensus map. And also the size of the feature maps. Everything will be annotated in the consensus map, as we will see in a minute. Then we just call the group function here. We pass in all our feature maps and the empty consensus map. The consensus map afterwards is not empty anymore. We also set the unique IDs for the features. And then we set the column headers, and we can save the consensus XML to a file. And that looks like this, also in XML format. And as I mentioned, the column headers are basically like this. So we have our map IDs enumerated, and then we have the MZML file names, and then we have the number of features that are derived from this specific feature map or this specific MZML file. But a nicer way to look at it is by using pandas data frames. And recently we've added that functionality in pyOpenMS. So you can use the getDF function to print out a data frame of your consensus map. And this looks like this, like a nice table. We have our unique identifier here and the charge, the RT, MC, and quality values of each feature, as well as the intensity from each sample. And yeah, what we can also do is we can plot it with Plotly with these 3 lines of code. And as you can see here, we filter also do some quality filter on our features. So it's not all of them, but we can nicely see that some features have a high quality here. It's also interactive. and some have a lower quality, and we can see their position on the two-dimensional map here. Yeah, and with this, I will hand over to Efi and for the bonus, which is about how you will generate the files for GNPS.
Yeah, so I hope you guys are still following, and it's not too much to go. So there's a few steps more in order to generate our nice directory with all the files necessary for feature-based molecular networking. So as Axel mentioned, we have annotated the MS2 data, and now we need to filter out the features that don't have these peptide identifications, right? So we introduced this input consensus that we generated from the feature linking step. And as already mentioned, we need to load this consensus XML to an OpenMS data file format. And that's the consensus map this time because it is a consensus file. So right after, we introduce a new consensus map, and that's exactly like our CMAP, the one that we loaded right now. But we only keep from that consensus map the metadata values. So we completely delete all the features. So it's like an empty vessel. So then for every feature in our original CMAP, in our original consensus map, we append the features that have peptide identifications in our new consensus map. So that's it. It's very easy. Pushback is like append if you work with Python lists. And then we just save the new consensus map in a consensus XML file as named filtered this time. So the next step is to export all the MS2 information, just cluster all the MS2 information basically in an MGF file, like we said. And we have an algorithm, a little class for that. So initially, yes, sorry, Axel, I'll just follow. So we are creating this directory called results GNPS export for just making our lives easier, basically. And here the script says that if it doesn't exist, then just make it, just create a new one. That's it. So then we introduce the consensus, the filtered consensus file that we just created. We also input all our NZML files from the example data directory. And as an output, we expect to get this MGF file with the MS2 information in the GNPS export directory. And that's what this class is doing. The spectral clustering, as we named it, is a GNPS MGF file class. It's just an algorithm. And if you run it with these inputs and outputs, then you get your nice MGF file that we can open up. Yeah, and it looks like that. So you have your feature ID information, your precursor mass information, the retention time, and then the list below is mass to charge information and also intensities of your fragments. Cool, and if we go back, there's two more files left. So that's the feature quantification table in a TXT format. So just to mention that all those three steps will be included in a single algorithm in the near future. And that's under development. I think we will have it in a few weeks, but right now we need to use another class and function to export our feature quantification text table. So what we need is our consensus map, our CMAP, the filtered one, and just an output file. That's the feature quantification table.txt. And all it does is like our Python script, basically converting the consensus map to a table. And it's a table that is in the right format for GNPS. Yeah, that's it. So you have your map ID information, the file names, and retention time, intensities, etc. Yes, and then finally, pandas data frames for your metadata table. So that's very simple. It's just, it parses through your example data directory and it just gets some information to generate the columns required for GNPS. And it's a little bit tricky. GNPS always needs this attribute in caps in front of all the columns except the file name. And yeah, also using those tabs is tricky when you do it manually. So it's nice to have the Python. Cool, yeah, that's it. And I want to thank you very much for today. Just before we finish, I just want to mention that we have, you can go for more information at this read the docs of pyOpenMS.read the docs. You can read all the summary and background information. We have installation instructions to import pyOpenMS very easily. And we also offer a lot of algorithms and workflows here for both targeted and untargeted metabolomics. Yeah, it's right here, exactly. And then for every single code that you see here, you can actually run it in a binder that you can see on the top right. Hopefully it works, but you can check later. Yeah, and I'm looking forward to hearing your questions.
Awesome. Thank you so much, Efi and Axel. This was great. And it's an awesome tool that I, till very recently, was not aware of is that nice. So maybe just a little context for like the frame of like our seminar. So in case you're excited now about pyOpenMS and you want to like use the binder that's basically readily available for you, and maybe you can also run it like offline with like with Jupyter or so. This basically perfectly plugs into feature-based molecular networking. So the files Efi generated at the end are like basically like the same files as Robin exported from MZMine. Just instead of like MZMine as a feature finding and pre-processing tool, you've now seen how to do it with pyOpenMS. And yeah, like in a scripting way, so like less with the graphical user interface. Yeah, this video is also on YouTube. So if you want to now take these files you just generated and then work from there, I think, I hope that you can easily do that. I think the only main difference would be that you have to select OpenMS as the feature finding tools than in the GNPS settings. And yeah, I guess with this, we're gonna move on to like the question and answer session in a second. And yeah, to those of you who gonna depart before, thanks again for joining, and I hope to see you next time when we actually gonna talk about how to visualize the molecular network and other data that you may create through the feature-based molecular networking workflow. So yeah, and also some more background. If you have not signed up yet, feel free to sign up to our mailing list so you can get the infos for the seminar and the Zoom links. We'll send this out every time before the lecture. And yeah, thanks again, Efi and Axel, for doing today's lecture. And yeah, looking forward to the discussion.